Hi, and welcome to this week's episode of Yan and Ben, the Biomen. Hi, my name is Yan Belolo, and today I will be presenting the cell cycle and mitosis. So, first of all, what is mitosis? Mitosis is the process of cell division where we create exact copies of each cell. So now, why is mitosis important? Well, when you were born, you started off as a zygote, and you eventually grew and became you through the process of mitosis. And so when a lizard will lose its tail or it will be cut off, it will regrow through a process also of mitosis. And we can also see mitosis here when we have scabs. They regrow also through a process of mitosis, and so mitosis is basically the process of all of how life and growth happens in our world. So because everything starts as one cell and eventually becomes a fully grown organism with billions and billions of cells. Before talking about mitosis, we have to know what diploid and haploid cells are. In eukaryotic cells, our DNA are organized into chromosomes. This would be chromosome 1, this would be chromosome 2, and this would be chromosome 3. This cell would be called a haploid cell because it only has one copy of each chromosome. Here, n would be equal to 3, where n is the number of chromosomes. And this one here would be called a diploid cell because it has two copies of each chromosome, and therefore 2n would be equal to 6. An example of a haploid cell would be a sperm cell and an ovum, where n would be equal to 23 for each of them. And after fertilization, they would combine and become a diploid zygote, where 2n would equal to 46. And so, after a lot of mitosis and development, that diploid zygote will become a fully grown human with 46 chromosomes, where 2n would be equal to 46. Therefore, it is a diploid organism. And so, you'd have 2 of chromosome 1, 2 of chromosome 2, 2 of chromosome 3, etc., where one of the chromosomes came from the mother and one of the chromosomes came from the father. Now, it is very important to differentiate between mitosis and meiosis. So mitosis is basically normal cell division, and it happens millions of times a day in all parts of the body as you grow and replace cells. Cells split into 2 daughter cells, where each daughter cell has 2 of each chromosomes, or diploid. And meiosis is the replication of gamete cells or sex cells. Mitosis produces 2 diploid cells, whereas meiosis would produce 4 haploid cells. In order for a cell to go through mitosis, there are 4 events that must occur. So the first one is called reproductive signal, which is simply the initiation of the cell into cell division. And then the cell will replicate. And then there is segregation, which is the distribution of the DNA into 2 new cells. And then there would be cytokinesis, which is the actual process of separation of the 2 new cells. We divide the cell cycle into 2 basic phases. The first one being interphase, and the second one being division. So interphase consists of gap 1, DNA synthesis, and gap 2, which is basically just the growth of the cell, the replication of the DNA, and more growth and preparation for division. Gap 1 and gap 2 are also known as G1 and G2, G standing for growth. And then there's the actual process of division or mitosis. As the cell prepares for mitosis, it has to go through a series of checkpoints. The first checkpoint during interphase is the G1 checkpoint. The second one is G2, which is also during interphase. And then there is the metaphase checkpoint, which is during cell division. The G1 checkpoint will check for cell size, if nutrients are all okay, if the growth factors are present, DNA is undamaged. During the G2 checkpoint, the cell will pass if the chromosome replication was successful and there is no DNA damage. And during the metaphase checkpoint, the cell will pass only if all the chromosomes are well attached to the mitotic spindle. As you can see over here, there is what is called the G0 state, which is for cells who don't copy themselves or cells who don't pass the G1 checkpoint. During the cell cycle, many mechanisms are in place to ensure everything goes okay. Cyclins are proteins that will regulate the cell cycle. Once a cell gets into the checkpoint, it will not continue unless it gets the signal that everything is okay and it can keep dividing. When one is okay, oncogenes can take effect. These are genes that will stimulate excessive division in the cells. This is usually regulated with tumor suppressors, which will stop or slow down the cell cycle. So, what is cancer? A cancer cell is a cell that will not respond to those stopping signals. And since they will divide uncontrollably, you can get a tumor, which is basically just an overgrowth of cells. The body, in order to stop tumors and cancer from happening, has many tumor suppressors. The main one is called p53. What p53 will do is it will stop the formation of tumors. When there is a malfunction or mutation in the p53 gene, it will essentially turn off the off signal and let cell division go ahead, and that will eventually lead to cancer.
DNA replication is where two identical copies of the original DNA strand are produced. The double helix unwinds and the two strands are used as template strands for a complementary strand by adding complementary nucleotides to the template strand to make an exact replica of the original one that you can see over here. Now we are going to talk about chromosomes, chromatids, and chromatin. The chromosome has an X shape, as you can see here, and a chromatid would be half of that, or one side. The other would be its sister chromatid, which would be over here, and they would be linked at the centromere, which is directly in the middle. Once all the DNA has copied itself, which occurs in the S phase, the DNA on one side, so we can say, let's say over here, will be exactly the same as it is over here. The DNA is folded over and over on itself until it is coalesced enough to look like a chromosome, which you can see here. Chromatin is when the DNA is still loose, which you can see over here, and is not fully coalesced to the level of a chromosome. Chromatin is where the cell is controlled. It's also where the protein is made. Chromosomes that are of the same size and have similar structure are what we call homologous chromosomes. Their genes are exactly the same and are in the same sequence, but their alleles are not always the same. In humans, there are exactly 23 homologous pairs of chromosomes. A lot of people always confuse homologous chromosomes with duplicated chromosomes. Duplicated chromosomes are two identical chromosomes that come from the replication of DNA, which are called sister chromatids that we saw in the previous slide, whereas homologous chromosomes are chromosomes that are one from each parent, as you can see here. There are four main phases of mitosis. The first one is prophase, followed by metaphase, then anaphase, and then telophase. In prophase, the chromatin has become coalesced in the chromosomes, right over here. The centrosomes move to the side, and the mitotic spindle starts forming, which attaches to the chromosome later on. The nuclear envelope breaks apart, and microtubules start to attach to the chromosomes, which form kinetochores. In metaphase, all the chromosomes line up on the metaphase plate, which is in the middle of the cell, right over here. The next phase is anaphase. In anaphase, they begin to be pulled apart to either pole, so each cell has the same genetic material, as you can see here. The last phase is telophase. In telophase, the nuclear envelope forms around both of these. The chromatin starts forming, the nucleoli appears, and the cells get much larger. As we can see, mitosis has occurred since the nucleus has divided in two. Once mitosis is complete, we have what is called cytokinesis. It is the last part of the cell cycle. When cytokinesis occurs, the cytoplasm divides in half, and you get two cells. Then, the cell will once again go through the cell cycle, which starts off with interphase, which goes through G1, and then S, then G2, and then followed by mitosis, where they will divide again. This is how an animal cell divides. It's a bit different for plant cells, which I'll show you in the next slide. Cytokinesis for plant cells is different than it is for animal cells. In plant cells, they form a cell plate that forms exactly between the two cells, forming a new cell wall leading to two cells.
Alright, so let's do a little quiz to see if you understood and remember what we learned today. Now, pause the video and click on your answer, and it will redirect you to see if your answer is right or wrong.